Thank you so much. I often say that the first Human Genome Project was such an amazing thing. We're just building on top of it. So it's nice to have that celebration toast at the end and share it with the Celera and the public team. Wonderful. So that's a great introduction because it's where I wanted to start my talk. It was really focusing on kind of perhaps a shared interest across the room and understanding that the human reference genome is foundational for most of genetic and genomic research today. And we can think about it outside of its kind of chemical components more as a coordinate-based system. All of these chromosomes, like chromosome 1, you can think of it as being chromosome 1, but every single base has a number associated with it. And this coordinate-based system, you can download wherever you are in the world, and you can begin to add information to it. So in a way, you can think about the human genome not as a chemical genome, but more like Google Maps, where the coordinates are like GPS. And you wouldn't use Google Maps if you didn't have all that rich annotation on top of it to let you know how you're going to get from point A to point B. And that is the same for the genomics community. Over 2 decades, we've spent billions of dollars around the world to make this incredible map. And this has driven a lot of new and exciting space, not only in basic science, but also in clinical genetics and genomics, and understanding our own human histories. However, as Mark alluded in this intro, the point I really want to make is that this is a tool. It's a tool that we've been utilizing for some time. However, it's not perfect. And we've known for 2 decades that this is an incomplete map. In fact, there's roughly 200 million bases that we knew we were missing. And that means all that annotation, all of that Google map can't be placed over these parts of our genome. So the biology here has also been missing. If we took this as a view from the first human reference genome, what your eyes can catch on to are these big red blocks. And hopefully you can tell by this figure that it's not scattered throughout the genome, but rather they're big red blocks that are collected on every single human chromosome. We know these gaps are really positioned over highly repetitive sequences, which I'll talk about. These are in areas like centromeres, acrocentric short arms, and over regions that contain segmental duplications. And this is important genome biology, because we know, like the centromeres I mentioned before, and you probably remember from basic biology, is a critical locus for how chromosomes divide every single cell division, whether you're in meiosis or mitosis. Understanding ribosomal DNAs, these are important for how we go through protein translation, but the site where these ribosomal RNAs are being transcribed have been missing from our maps, as well as understanding the segmental duplications that I'm showing here as kind of being markers, as you can see on the 2 different chromosomes that are blue, where you have pieces of your genome, which are then copied and pasted almost in terms of having a copy that's present somewhere else in the genome. And these are sometimes attributed to genome instability of these expanding gene families. So as mentioned, I'm here representing one of hundreds of people who came together with a call to action called the Telomere-to-Telomere Consortium. And our goal was to try to create the first complete end-to-end map of a human genome. And so in this case, that kind of chromosome cartoon that I was giving you before, we're trying to create what we call one contig, or one assembly, which would have a telomere in one end, a telomere in the other, and high accuracy bases all the way across. We were successful in this, and we celebrated back in 2022. Here you can see the group of us that are getting together. This is the first time we actually met in person. This was after the pandemic. And it was really at this point that we were able to dive into some of the new biology that I'm going to talk about today. Now, it was not just the people who came together, the development of new tools, as you can probably benefit from even the earlier sessions when we had that lovely introduction by EpiCypher. It's really this rise of long read technologies that presented this opportunity. And so I wanted to go through, since this is meant to be more of an instructional-based talk, some of the newer long-read technologies that enabled this type of progress. The first one is known as hi-fi sequencing. This is from Pacific Bioscience, or PacBio. And you can see from this slide, it's kind of like a circular consensus sequencing where you have kind of a circular DNA. If you have a polymerase that goes round and round, you can make several subreads. And it really is taking the consensus of across all of these various subreads that gives you an incredibly high fidelity or high quality read. Some people equivalent this to Sanger reads in terms of having such a high quality. In addition to this highly accurate read, we also made use of ultra long sequence data. This is from nanopore technologies here. This is the protein which is embedded within a pore. And you can have a DNA pull through. And in this case, the DNA length is really dependent on how you can how long the DNA actually reaches the pore. So if you present the pore with 1 million bases, it can read 1 million bases. And so a lot of effort went into carefully preparing this high molecular weight DNA, the delivery of this molecular weight DNA, in order to deliver these extremely or ultra long sequences, which are typically defined as any read that's greater than 100 kilobases in length. As I'm showing you here, you can see it's not just 100 kilobases. That might be where we draw the line with how we use that definition, but there's a long tail that extends out, sometimes going even into megabases. So long reads were important for solving duplicated sequences. Here I'm just showing you an example where you could have a blue sequence in the genome, and then it's duplicated to be the light blue. And there could be mutations that acquire differences or present differences between these two copies. Once again, I'm trying to show this more as a lightning bolt, where you can see in the light blue, there are two site differences that differentiate it from the dark blue copy. So if you have short reads, in order to be able to tell the difference between these two copies, you have to be lucky and kind of overlap these very distinguishing sites. However, when you have longer, more accurate reads, you're able to increase the likelihood of overlapping some of these informative sites. And of course, this becomes very easy to do when you start to work with these much longer, ultra-long sequences as well. And so it really was this idea, if you're starting off with a puzzle metaphor, which is what we normally do for genome assembly, and you're now working with some of these blue sky pieces of the puzzles, or the parts of the puzzles that they all are the same color, they look like they're in the same region. If they're small, it's very difficult to put together. But of course, if you have, versus 2,000 piece puzzle or 200 piece puzzle, it's a lot easier. And this was what the real advantage was for utilizing some of these long read technologies. Now we were able to actually implement a hybrid approach to put a human genome together. And so here you're going to start off with those extremely high quality or high accuracy reads, or the HiFi. And we're going to use a software known as Verco, which was developed by Adam Phillippe's group, which I'm highlighting here. And so you can take the HiFi data sets and you can begin to go through and find stretches of the HiFi that are identical to one another between the read data sets and begin to build the first string graph that I'm highlighting here. You can utilize the ultra long sequences. In this case, intuitively, you can use these long sequences to begin to thread through or identify the paths that exist in this high accurate string graph that we created at the beginning. And then you're going to want to know which of those paths belong to the maternally inherited complement chromosome and the paternally inherited complement chromosome. And we do this many different ways, but the ones I'm highlighting here is we use a trio-based approach. So in this case, if you were trying to build a T2T genome from an individual, you could use their parent's DNA. And using the parent's DNA, you can begin to tease apart or phase the two string graphs I'm showing you here into the two different paths to bring up the two T2T chromosomes. And so when we first started the Telomere-to-Telomere Consortium, a lot of this was kind of being built by amazing engineers and people, and Adam's team, to really give credit to Sergey Nook in particular and Sergey Cohen, who's on this slide. But now we've been working as a consortium to really start to automate a lot of these tools to ensure it could have broader use. Now, of course, when we think about the original Human Genome Project, which I'm highlighting the first submission being HG4 down at the bottom, and the release year being about 2001 when it first came out, now we can start seeing that the T2T genome is really a step change in terms of the number of bases that are out, and it really was something that was different in terms of quality. So what was introduced in this new T2T genome, and how is this going to impact perhaps some of the studies if you choose to use it? Well, without question, these parts of the genome, as we already talked about, were repeats. And we're probably all very used to thinking about repeats here. I'm just taking an image from Wikipedia where we're starting to think about things like sign or allo repeats, where we know they represent about 10.6% of a human genome. And if we were able to label that repeat and actually show it to you as a kind of a fish on top of chromosomes, you'll see a lot of these various chromosomes just light up to really give you that feel of how repetitive our genomes are. And so if now we're thinking about unique sequences, as I'm showing in a cartoon down below, as being green, unique being green and the repeats being red, you can see the repeats are more or less any sequence that's found in multiple copies in a genome. And if we think about this in a very kind of basic way of a genome being like a book, then you can consider an interspersed repeat kind of like being a short phrase, like the dog. Here is the dog chased a squirrel, the dog jumped over a fence. You can imagine where I'm going. This is kind of interspersed throughout. So these type of interspersed repeats, just like the allo element, was one of the discoveries that was released by the T2T Consortium in our CHM13 genome. And this is really crediting some work from Rachel O'Neill, who's here at UConn, and her student Savannah Hui, where they were able to run repeat modeler and actually predict 5.5 million bases of new repeat predictions and begin to map them in the context of this newly released genome. Another type of repeat outside of this type of interspersed repeat is one where you might imagine that book metaphor again, but instead of just having a short phrase that's kind of reused over and over again, here you might have an entire paragraph of that book copied, and you'll find it a chapter later, the exact paragraph again. And this is how we can think about things like segmental duplications. Here, these are blocks of near identical. We're looking typically around 98 to 100% identical of DNA that can range anywhere from 1,000 to 400,000 base pairs in length, and they contain many genes. And these can vary between individuals. And so the gene copies that are sometimes inherent in these blocks also can vary across individuals. And this is really giving credit to a lovely study that came out of the T2T Consortium, and this was led by Mitchell Volger and Evan Eichler at University of Washington, where they were able to really classify all of these segmentally duplicated events, and they found that they could identify 81 million bases of these events that existed in the CHM13 genome, which is roughly 7% of a human genome. And what I'm using the slide to show you is, if you see in red, these are all the genes that were present in CHM13 that were missing from our prior human reference map. You might say, but wait, there's some genes in black. Those are ones that are shared with our prior human genome reference, but they were in a misorientation or reorganized where they didn't have a match with CHM13. And so now we're kind of building this narrative. Now I'm going to share with you satellite DNA, which is closest to my heart, because this is where my lab has been putting the most research in for the past decade. And so in this case, you can imagine the dog being an interspersed repeat or even a copy of a paragraph, but here you almost have the same word written over and over and over and over again for pages and pages. So this is the idea behind satellite DNAs, where sometimes you have a stretch of DNA, which can be found in a head to tail orientation, is typically repeated over and over again for tens of millions of bases on some chromosomes. And we find that these are not randomly distributed. The satellite DNAs are typically in heterochromatic regions and areas like the primary constriction, like on human centromeres. So my team and the SynSat team from the Telomere-to-Telomere group actually led the first maps of the human centromeric regions. Here, I'm just trying to give you a feel for it. Here, what I'm showing you in red, hopefully your eyes can see that on every single chromosome. These are the sequences that typically serve as a substrate for these centromere kinetochore proteins that come in and actually ensure proper chromosome segregation. However, there are some really massive satellites here as well, like your eyes may catch on chromosome 9 or chromosome 1. These are ones that we've never seen in prior human reference genome maps. And so it presents a whole new biology to try to chase down what these satellites are doing, how they're different between individuals, and how they contribute to genome biology. Now, I know when I talk to individuals, I could probably spend a whole hour talking about the satellites and what they could be doing. But I do often get this question as well, is are there genes that are actually hiding in these new areas? And the answer is yes. So we were able to team up with some scientists. Here I'm highlighting Mark Deakins, who's also from UC Santa Cruz and participated in the GenCode project, and Savannah Hoyt, once again here from UConn, where we went through and we were looking at not only the predicting genes, but also looking at the transcription through these sites. Okay, so good biology starts with the great genome. This allows us for variant calling, RNA-seq, gene and repeat annotation, and chromosome structures. And if you have an incorrect map, that leads to incorrect interpretation if there's a take-home. And so utilizing these complete maps, now we can start to expand comprehensive variant discovery. We've made new discoveries by taking data sets like 1,000 genomes and mapping it back to the CHM13, identifying hundreds of thousands of novel variants per sample and identifying samples that present error, false calls. We've been able to identify new biology from these maps, gone through and done careful annotation, pointing out expressions and sites of methylation and new repeat classes. This is just providing a snapshot of looking at an acrocentric short arm. We can begin to make hypotheses now, thinking about things like Robertsonian translocations, where we expect the short arms to actually fuse and be one of the highest structural incidents that happen in the human population. We can look at these maps and we can start to find trends. For example, on 13, 14, and 21, matching some of the Robertsonian literature, which shows that these are more frequently involved in these types of structural rearrangements that are also seem to be highly duplicated and in different orientations. And finally, we can start to use these types of methylation. I think this was introduced earlier in the workshop as well. If you're reading long reads through, it could be HiFi or it could be the ONT platform, you're able to actually predict the DNA modification of the CPG. And so when you're mapping reads back to this genome, now you get this added epigenetic information as well. And if you start looking over some of these sequences, like here I'm highlighting centromeres we've never seen before, we're starting to find some really cool new epigenetic signatures, like these dips that we know are associated with centromere proteins. So now we're moving into a world of future advancements for T2T technology. If you're here for the rest of the week, I'll spend my next talk really talking on these production, how we're pushing things to go to hundreds and thousands of T2T genomes. This is where we're in a very excited space. And the reason that we want to do this is because there's a lot of new unexplored variation, not only in the centromere, which I'm highlighting here, that have different sizes, the gene families, understanding how they expand and contract, or even whole acrocentric short arms, which we know have evidence out there swapping between individuals. And even looking at epigenetics, we can start to see trends of where they're starting to be in one position on a centromere and one individual on another. And we have no idea yet how this contributes to how the chromosome function and how they segregate. So with that, I'll end my talk. I'll be happy to take any questions, but I just want to emphasize that, these T2T genomes are going to be really important, and a lot of interest is going into understanding how a complete variant study can help us better understand healthcare. We're now applying these methods to begin to look at comparative genomics. There's opportunities to look at human evolution, as well as thinking back into non-human primates and to other mammals. And we're looking into new cell biology. So with that, I'll go ahead and once again express my gratitude for this amazing team who came together, and I'm presenting on behalf of today, and I'll be happy to take any questions you have. Thank you.
Hi, Karen. Lovely talk. So I have a question about, surprise, the link between DNA methylation and centromeres.
Mm-hmm.
So because TETs and DNA methyltransferases are mutated in various cancers, and you're now seeing DNA methylation at these centromeres, could you comment on what's known about the impact that those mutations would have? And then also, conversely, the therapies are targeting DNA methyltransferases. So what do you think is going on at centromeres in those contexts?
This is a really exciting space, and I know a couple of labs are actually trying to test this now, so I can't release too much. But I will say that the interest here is actually understanding what that importance of the CDR is. So there are some models where the CDR is just an early development signature. Oh, I'm sorry, the centromere dip region. Thank you for helping me clarify that. So when you look over the centromere, you have that methylation profile, and then you rightfully know there's a dip. And that looks like it has a methylation signature that could be important for function. And I think that's a really exciting space. I don't know of any literature yet that's going into great detail of studying that in cancers. I know a lot of people are in this space right now, so stay tuned. But I do think that what I'd like to answer you with is we don't know how important that actual CDR is for maintaining the centromere, because we can find, at least for the published work that's out, sometimes you find centromere proteins that are outside of the CDR. It's not a completely one-to-one ratio. So you could imagine a hypothesis when you're in cancers and you now expanded or contracted that CDR, you now have to test a hypothesis that might modulate the kinetochore, or it may have nothing to do with it. And it might just be that that's an early developmental mark, and it's just kind of telling us that's where the footprint was when things got set up. And maybe it doesn't have such an important role in somatic cell development. That's one model. The other one is that it's driving it. And so if that model is correct, cancer would be an incredible place to look, because that's where we expect to see that type of heterogeneity. Yeah. Great.
Okay, let me ask a question, which is probably not a simple one, but I'm going to try. So one role of duplication is to create multiple copies of genes that can evolve independently to new functions. So creating diversity by gene duplication. At the same time, ribosomal RNAs are among the most conserved sequences in the genome, and they're massively duplicated. How come those don't diverge into oblivion?
Well, I think that we do find a lot of divergence inside of those, interspersed, or the ITS regions on the ribosomal, which is how we've been mastering all these phylogenetic trees and having differences between. But the actual purpose of where the ribosomals or RNAs are focused is really to create this highly conserved ribosome. And so you can imagine that's what's keeping that, kind of in check. And of course, you can imagine that having just you have to have a huge number of these types of ribosomes. And so something has to be driving in the genome, both ensuring conservation, which is the homogenization, as well as repeat expansion. And so it's really kind of a cool idea of what, knowing that that has to happen in order for that species to be successful, especially in early development, when you need a huge amount of ribosomes and you need them not to be broken, right? I think will be very interesting going forward as we start to look at this mechanism and how it's conserved between different species going out.
Hi, very nice presentation. So I was very surprised by your explanation that you were able to sequence the chromosome from one end to the other end. And in the pictures we always see the DNA or the chromosomes as a unit with a middle part or a centromere part. So my question is the chromosome a single piece of DNA, and that centromere is like a region that is part of the DNA, but is also having proteins together there? What made the centromere, and does it encode genes as well, or it doesn't have genes, it's just a structural part of the DNA?
There's a lot of questions in there. I'm first going to make sure I say this correctly. No one that I'm aware of can sequence a chromosome end to end on one sequence read. This takes long read, high coverage sequencing and assembly mechanisms to put together a contig. And the contig is an assembly product. And so at that point, once you have an assembly product, which is composed of reads, then you have that one chromosomal length type of sequence that you can work from. Now, where is this? One DNA strand. Exactly, but it's a assembled sequence. Yeah, as long as that's clear. And then the question about how we know it's a centromere, right? I think that's an incredibly important one that sometimes we gloss over, and that some of the literature has suggested that centromeres can pop up in many places in your genome. They actually have some flexibility, and sometimes they can pop up even over areas that contain genes. So we don't really have a completely 100% a sequence is going to drive a centromere's location. What drives a centromere's locations are these key centromere proteins and where they are embedded within the DNA is being wrapped around them. And those are the signal or the beacon that say, hey, kinetochore assemble here. We just know that if I was a betting person, which just say I am for fun, I would take everyone's genome here. I feel very confident it's all going to be over this alpha satellite that I showed you, because of all the clinical work that's been happening for decades. We've been mapping alpha satellite and mapping that primary constriction that you just referenced. And most of the time it's over these sequences. And in the past, what people did is they took subsamples of these sequences, they made sure they had no proteins on them, created a circular vector, like a human artificial chromosome, put them back inside of cells, and demonstrated that there's something unique or interesting about these sequences because they're capable of recruiting those important proteins de novo, right? And creating a centromere on this type of artificial construct. And so because of that, there's also some motivation in the field to understand the genomic association with centromere function too. I hope that answered your question. Thank you.